California coast is one of the most biodiverse marine environments on the planet. But beneath its surface, something is changing. Blooms of microscopic algae, invisible to the naked eye, explode in the population, discoloring the water. These are harmful algal blooms, and they're more dangerous than they look.
Today, we will be using statistical analysis to explore how environmental conditions like temperature, salinity, and dissolved oxygen correlate with phytoplankton abundance along the distinct regions of the California coast.
The warm, stratified waters of Southern California and the cold, upwelling-driven waters of Bodega Bay.
The question driving this investigation: statistically, how does the abundance of HAB-forming phytoplankton species correlate with environmental and biological variables in California coastal waters? To understand harmful algal blooms, we first need to understand what they are at a microscopic level.
Phytoplankton are microscopic organisms that form the base of the ocean food web. Most are completely harmless, essential even. But certain species, under the right conditions, can multiply rapidly and produce toxins devastating to marine ecosystems and human health. Among the most concerning are Pseudonychia, which produce domoic acid, and Dinophysis, responsible for diuretic shellfish poisoning.
When humans consume contaminated shellfish, the consequences can be severe.
HABs cost the U.S. seafood industry tens of millions of dollars annually and have been linked to the deaths of sea lions, dolphins, and seabirds along the California coast.
3 key ingredients:
nutrients, particularly nitrogen compounds, warm, stable water, and sunlight. When these align, phytoplankton populations can double in a matter of hours. But not all parts of the California coast are equal. The physical oceanography of Southern California and Northern California are fundamentally different. In Northern California, upwelling dominates. This is where winds push warm surface water offshore, and cold, nutrient-rich water rises to take its place.
To answer this question, we needed data. Real, reliable ocean data, collected not from a lab, but from the ocean itself.
Meet the Spray Glider, an autonomous underwater vehicle about the size of a surfboard that cruises through the ocean on its own, collecting measurements every few hours for months at a time. These gliders are part of CUGN, the California Underwater Glider Network, operated by Scripps Institution of Oceanography. In my case, 4 variables were measured: temperature, salinity, chlorophyll, and dissolved oxygen.
Chlorophyll deserves a special mention. Counting individual phytoplankton cells across hundreds of kilometers of ocean is basically impossible. So instead, scientists measure chlorophyll A, the green pigment inside phytoplankton, as a stand-in for how much phytoplankton is actually there. More chlorophyll means more phytoplankton.
To analyze the data, this study used correlation analysis. The result is a variable called R, ranging from -1 to +1. +1 means the 2 variables rise together. Negative means when one goes up, the other goes down. The closer to 1 or -1, the stronger the relationship. The closer to 0, the weaker. Think of it like this. If you measured ice cream sales and temperature on the same days, you'd probably get a stronger positive R value. Hot days mean more ice cream. That's a positive correlation. This study uses the same logic, just applied to the ocean.
6 correlations were tested across 2 regions. These correlations came back statistically significant, meaning the relationships found are real. Here's what the data revealed.
In Southern California, temperature is the single strongest predictor of phytoplankton in this entire study. The correlation is R negative 0.654. It's strong and statistically significant. Every degree of warming pushes chlorophyll down. The Bay Area returns R positive 0.276, weaker, but pointing in the exact opposite direction. Here, warmer water is associated with more phytoplankton, not less. Same variable, same test, opposite sign. That single contrast tells you more about the difference between these two systems than almost anything else. Salinity reinforces a similar story. In Southern California, saltier water seems to correlate with more phytoplankton. The R here is positive 0.561. In the Bay Area, the relationship flips. Fresher water correlates with less phytoplankton. The R here is negative 0.320. Here, fresh water signals coastal mixing and runoff, delivering nutrients to the surface through an entirely different mechanism. The confidence intervals for the R values don't overlap, statistically confirming that these are genuinely 2 different systems.
Dissolved oxygen flips the question. Instead of asking what drives phytoplankton, it asks what phytoplankton does to the water around it. Phytoplankton photosynthesize, releasing oxygen. So more phytoplankton should mean more dissolved oxygen. And that's exactly what these tests show.
In Southern California, the R value is positive 0.336. In the Bay Area, R is positive 0.604. It's nearly twice as strong. The Bay Area's upwelling system is simply more biologically intense, and the oxygen signal reflects that.
One important thing to note is that this measures living blooms producing oxygen. When a bloom dies and sinks, bacterial decomposition consumes oxygen in massive quantities, creating hypoxic dead zones where marine life suffocate. This destruction comes after the bloom, not during.
Plotting chlorophyll over time reveals the biggest contrast of all. In Southern California, the pattern is dramatic, but unpredictable. Chlorophyll suddenly spikes, particularly in spring, shooting up to 12 times its baseline before crashing back to 0. The Bay Area looks fundamentally different. Yes, intense spring spikes do appear, particularly in 2022, but between those spikes, chlorophyll never fully crashes. A persistent baseline of 1 to 2 milligrams per cubic meter holds through fall, winter, and through the quietest months.
That floor is the fingerprint of upwelling. Even when seasonal winds weaken, cold, nutrient-rich water keeps reaching the surface. The Bay Area's ocean is never starved the way SoCal's is. Hypothesis testing was also used to determine statistical significance. A p-value measures the probability that the observed relationships happen by chance. A p-value below 0.05 indicates the relationship is unlikely to be due to random chance. In this study, all p-values were below 0.05, so the null hypothesis of no relationship was rejected in every case. This provides strong statistical evidence that the relationships observed are real. So what does all of this tell us about harmful algal blooms in the California coast?
Well, the data reveals 2 oceanographic systems that, despite sharing the same coastline, operate by fundamentally different rules. In Southern California, the physical environment, specifically temperature, is the dominant force controlling phytoplankton abundance.
In the bay, no single variable tells the whole story. Temperature and salinity both show weaker, opposite relationships with chlorophyll compared to SoCal. This isn't a flaw in the data, it reflects the complexity of an upwelling-driven system where nutrients, mixing wind, and biology all interact simultaneously. Upwelling is not one variable, but it's many variables working together. These regional differences have real consequences. Northern California's upwelling-driven productivity creates the conditions for more frequent and intense HAB events. The same nutrient-rich cold water that makes Monterey Bay one of the most biologically rich marine environments on the West Coast also fuels the pseudonychia blooms that poison marine life with domoic acid.
Southern California faces a different kind of HAB threat. Without the strong upwelling to drive productivity, SoCal blooms are more often tied to nutrient runoff from urban development, stormwater, agricultural discharge, and wastewater carrying nitrogen into coastal waters. Temperature remains the key variable, but here, human activity plays a larger role in supplying the nutrients that temperature alone cannot.
What statistics gives us is a way to quantify these differences objectively. Not just to say that the Bay Area is more productive than SoCal, but to show precisely how strongly temperature predicts phytoplankton in each region, how differently salinity behaves, and how much more tightly biological activity tracks oxygen in an upwelling system. Numbers give the comparison meaning. So, statistically, how does the abundance of HAB-forming phytoplankton species correlate with environmental and biological variables in the California coast? The answer is clear. Environmental variables do correlate significantly with phytoplankton abundance in both regions, but the strength and direction of those relationships differ fundamentally depending on where you are along the coast. In Southern California, temperature is a strong, reliable predictor of phytoplankton abundance, explaining 43% of chlorophyll variation with a definitive negative relationship. Warmer water means less phytoplankton.
The ocean here is governed by thermal stability. In the Bay Area, no single variable dominates. Temperature explains less than 8% of chlorophyll variation and points in the opposite direction. Salinity flips too. Instead, phytoplankton abundance more strongly drives dissolved oxygen, reflecting a system where biological activity fueled by upwelling is the dominant force.
The ability to measure relationships between environmental variables and phytoplankton abundance gives scientists, policymakers, and communities a more powerful tool for anticipating HAB events before they happen. But the stakes are real. Harmful algal blooms threaten food security, marine ecosystems, and coastal economies. Understanding what drives them statistically, regionally, and oceanographically is a necessary step towards protecting the California coast for the communities and wildlife that depend on it.
The ocean is complex. No single number captures it fully. But statistics give us a language to describe it, to find patterns in the noise, to compare places separated by hundreds of miles of coastline, and to ask better questions about a changing sea.
Reducing harmful algal blooms requires actions on multiple fronts. It's reducing nutrient runoff from agriculture and urban development. It's expanding real-time ocean monitoring networks. It's supporting continued research into the complex drivers of HAB formation. And it's communicating this clearly to students, policymakers, and the public. Because the first step to solving any problem is understanding it.